Welcome to the Ketogeek podcast. Let's do this.
Health, nutrition, fitness, low-carb lifestyle.
Hello, everybody. Welcome back to the Ketogeek podcast. My name is Fahad, and today's show will be solo hosted by yours truly. Corey is a little occupied today, and I decided to take the helm and delve into this Pandora's box. Today's topic is very intense, very science-heavy, and but we're going to parse it down. We're going to try our best to keep things as simple as possible. You got to realize we're talking about stuff that a lot of PhD researchers are throwing our way, so there is going to be some level of complexity, but we'll do our best. And explain to you guys what's going on. But before we start the show, let us talk about Ketogeek. Ketogeek, for those who are uninitiated, Ketogeek is a food business, and we also do this podcast that you guys are hearing. We like to stay on top of the latest science and knowledge, but we express ourselves in these beautiful 2 products that we sell on our website, and there's some more good news coming as well, besides our website. Well, one of the products that we sell is ghee that we make, and we have basically gone throughout the entire country, tried to find the best butter we could find, and we heat treat it use the traditional method to make ghee from it, and then we packed them in these glass jars. We don't use plastic at the moment. We're looking into it, but glass seems to be the safest at the moment, so we're using them. And we sell them on our website at ketogeek.com. Alongside, we take this same ghee and bunch up other high-quality fats. We use MCT oil, and we use almond flour, and we use sugar-free sweeteners, and some other things to create these tiny little cups called energy pods. And they are also available on our website at ketogeek.com. These are shelf-stable products that you can take anywhere you want. We've improved their texture. They are also more shelf-stable and resistant to separating. That was one issue we used to have with the energy pods. They would separate after a few months, and now they're quite resistant to it. And so we're happy. We're happy where it stands. And how do you consume them? Well, you can treat them like a pudding if you want to. I like to tag them alongside a high-protein meal. I could consume steak or chicken or any of those kind of things, and then I'll put an energy pod right next to it to add some extra fat to it. If you're adding fat, why not make it tasty? And plus, one of the best things about going on keto or low-carb, well, that's one of the earlier attractions alongside all the science was I could have chocolate every day. So the chocolate fudge energy pod came into being, and so shared with a bunch of other people, and then it just turned into a product that everybody likes. And we just went in stock after a year and a half, so we opened the floodgates last Thursday, and a lot of you people ordered it up, and I really cannot express my thanks to everybody who has ordered, and some people ordered many of these boxes, and each of these boxes that you buy contains 8 of these pods, and each pod is about 2 ounces of good stuff. That's what I have to say, good stuff. But the way I consume sometimes, besides adding it to protein, is I'll just be working and have an energy pod open right next to me, and perhaps maybe some caffeine alongside, and I'll just take the spoon, the paperboard spoon that comes with it, and just eat a little bit, like a tiny spoonful of it, just to get that little dose of chocolate goodness that I crave from time to time. And then I'll eat the whole pod throughout the entire day, just one pod. Sometimes if I'm really hungry, I'll eat just 2 of them, but I prefer to keep it to 1, which is one is good. A single number is good. So yeah, energy pods are available at ketogeek.com, and there's good news that I mentioned earlier. We will be on Amazon as well. We're opening those floodgates pretty soon. They won't be on Prime yet. We're still figuring out the logistics before we go into Amazon Prime, because Amazon Prime is a little bit more complicated. You have to send Amazon the product, and then it goes into their whole distribution network, and it slowly trickles down to your local facility or Amazon shipping facility or distribution center, and that's how they're able to offer you the 2-day shipping. or sometimes even the one-day shipping, because they're so distributed down to your local center that it's very easy for them and really fast for them to ship them to your doorstep. I think their model's kind of really cool. So that's it when it comes to energy pods. Go buy them. They really support our podcast. Whatever you're hearing over here is 100% fueled by energy pods and our ghee. So the more you guys buy them, the more we can churn out good content, the more we get the more time we get to invest in quality information. We got some big plans out there. We can't do them without you and your support. And while you're at it, leave a like or a review for this podcast and our product too. That really helps as well. Gets it to more people out there. Means a lot. So really cool stuff. Now, going back to the original topic, we started our journey last week with the metabolic response when it comes to macronutrients and energy intake. But this week, we're going to delve into the endocrine response, and this is going to be a little bit more complicated. It's a next level, a little bit of a higher tier of knowledge. I wouldn't say you should be intimidated by it, but I would say that you may have to listen to it more than once to understand what is going on. It's not that hard, and even if you don't understand what exactly is going on, try to abstract these little golden nuggets of information that seems relevant to you, and perhaps you can, in the future, come back to the show again. And this podcast is mostly based on this 2016 review paper called The Macronutrients, Appetite, and Energy Intake, which will be referenced in the show notes as well. And I mentioned it in the last show as well. One of my favorite researchers is Dr. Richard Mattis, and his team is phenomenal. They were the ones from Purdue Nutrition, I think. They wrote this paper, and I love it. So don't be too intimidated. And sometimes the best knowledge you can acquire are things that you cannot easily observe. It requires some critical thinking. That's how humanity has pushed the limits of understanding. You gotta go beyond the obvious. You gotta search for paradoxes. You have to actively falsify. A lot of things that we knew at Ketogeek eventually became falsified by us. So I invite all of you to this journey, and it'll be a good time. We are going to get into some complicated topics, like I said. But I want everybody to know that please pardon my pronunciation, as I'm mostly a reader, and we gave this warning last week as well. So if you really want to make fun of my pronunciation, please laugh at it and leave some comments, and perhaps correct me on it wherever I'm wrong. I didn't take biochemistry as a major as well, so. But with all of this being said, let us delve into this amazing topic and have some fun. First things first, let's talk about what exactly is the endocrine system. And so the endocrine system is a collection of glands in our body that produce hormones that regulate metabolism, growth, development, tissue functions, sexual function, reproduction, sleep, mood, and other things. A hormone, on the other hand, is a chemical messenger secreted directly into the bloodstream and is then transported to organs and tissues of the body. They slowly affect these aforementioned processes. These glands or organs in our endocrine system are located in various places, such as the hypothalamus, the pituitary gland, and pineal gland in your brain, adrenals on the top of your kidneys, pancreas behind your stomach, and ovaries or testes in your pelvic region. The endocrine function specifically for the gastrointestinal tract or the GI tract was recognized as early as the work on secretins in 1902. Secretin is a hormone released in the bloodstream by the duodenum, and the duodenum is the upper small intestinal tract right outside the stomach. So it's kind of like that small little piece right there. Yeah. Digestive processes are purported to be controlled by these gut hormones or peptides, and the list of these peptides has grown to over 20. In case anybody's curious, peptides are just simply small chains of amino acids. Very, very simple, but yeah. These hormones or peptides are secreted by 15 to 20 different types of enteroendocrine cells, characterized by the peptides they release and their anatomic location. Enteroendocrine cells are special cells that are found in gastrointestinal tract, stomach, pancreas, and that they respond to nutrient ingestion. Henceforth, the GI tract is now viewed as the largest endocrine organ in the body. One of the hormones in the context of this topic is cholecystokinin, or CCK, which is a hormone secreted in the duodenum and stimulates the release of bile into the intestines and the secretion of enzymes by the pancreas. One of the earlier discoveries on CCK, or cholecystokinin, was its impact on gallbladder contractions, demonstrated in 1928. However, its influence on energy intake or meal size in rodents was not established until 1973. This finding prompted the research for other peptides that modulate appetite and ingestive behavior, with characterization of more than a dozen hormones such as CCK, PYY, GLP-1, GLP-2, OCM. There's a lot of them. We're not going to delve into them. So some of them held anorectic properties, such as appetite suppressing properties. And there was this one called ghrelin, which was orexigenic, or fattening, or appetite stimulant. Yeah. These satiety hormones are secreted by enteroendocrine cells, termed open because they have a microvilli exposed to the interior of the gastrointestinal tract that contain receptors for nutrient signals derived from ingested food passing through the GI tract. Microvilli, just to give a side note, is a plural for microvillus, are cell membrane protrusions that look like tiny little bristles in a microscope, which increase surface area for diffusion and aid in the absorption, secretion, and other cellular functions. These nutrient receptors are located on I cells in the proximal or closest, and L cells primarily in the distal or farthest part of the GI tract. Personally, I like to keep things very simple, and I say I cells means I for initial, and L cells mean L for later or lower, whichever works for you, whatever floats your boat. And then, of course, GLP stands for glucagon-like peptide, and PYY is short for peptide YY or peptide tyrosine. We're just going to keep things simple here, and we will call them GLP or PYY, because they're just a whole new can of worms if we delve into the depth of knowledge when it comes to these hormones specifically. Anyway, L cells are also present in the duodenum. That is the small intestine area right outside your stomach. I'm going to repeat certain things occasionally just so that we stay on track, and some people want to do some catch up. So don't mind me, especially if you already know all of these things beforehand. I'm sorry about that. Other enteroendocrine cells, such as P/D1, not to be confused with PD1, which means programmed cell death protein 1. So the P/D1-like cells in the upper part of the stomach that secrete ghrelin are closed. Those were open, this is closed. And the closed don't likely monitor the nutrient mixture in the gastric lumen. And as a side note, P/D1 cells are the ones that are removed during gastric bypass surgery. And there are issues with the gastric bypass surgery as well, but that's a topic for another day. So ingestion of monosaccharides, or simple sugars, and complex carbohydrates are associated with ghrelin secretion, but this is probably not directly responsive to nutrient signaling, as closed forms of enteroendocrine cells respond to neural and hormonal signals. The initial work on endocrine signaling in the GI tract focused on the stomach and small intestines, but more recently has expanded to range from the oral cavity or mouth to the colon. The promise that manipulation of these modulators of feeding and glucose tolerance could be harnessed to address the growing overweight obesity pandemic and the diabetes problems identified in the late 1970s in the US prompted research that expanded understanding of gut biology but has not yet yielded hope for clinical results. The oral sweet taste receptor, first characterized in 2001, binds an array of sugars as well as low-calorie sweeteners. In addition to providing a sensory signal that is inherently pleasant and guides food choice, Rodent taste cells that bind sweeteners also express GLP-1, which we discussed or touched a little bit earlier, which stands for the glucagon-like peptide, which is a hormone produced in the L or the later cells of the distal region. GLP-1 may be involved in sweet taste signaling and may also enter the circulation, where it could exert a direct anorexic or appetite-suppressing effect on the brain. The concentration of GLP-1 released from the taste cells is limited, but because there is little or no enzyme present to degrade the molecule, as occurs following the release from L cells in the distal intestine, it may exert a larger effect than expected. One of the terms that you guys are going to just get exposed to, or some of you probably already know, is the cephalic phase insulin response. And what do we have to say about that? Well, these guys, what do they have to say about that? The paper writers or authors. The cephalic phase insulin response, or the anticipatory release of insulin due to sight, smell, and taste of food, has been traditionally ascribed to vagal-mediated pancreatic secretion. Vagal basically means it's attributed to the vagus nerves, whose one major function is to bring sensory information from organs such as tongue and ears to the brain. However, recent findings suggest that there may be also an endocrine contribution or component via taste cell release of GLP-1 we just talked about. Alternatively, a signaling system activated by selected sweeteners independent of the sweet receptors may be contributing. Such a system has been described in mice, but its existence in humans is not known. Okay, the cephalic phase insulin response is significantly correlated with post-absorptive insulin concentrations and has been associated with ingestive behaviors. However, the roles of glucose and insulin in mediating appetite and feeding under physiological condition that is realistic without absurdly impossible doses that the body itself cannot produce are questionable. In 1958, it was reported that the presence of nutrients in the ileal lumen, that is, the last part of the intestines, inhibits gastric motility and enhances satiation. In the early 1960s, gastric infusions of isocaloric, meaning same calorie, test solutions of glucose and starch were reported to slow the rate at which food leaves the stomach and enters the small intestines. This rate, of course, is called gastric emptying. Subsequent work revealed that glucose ingested orally through your mouth or administered gastrically had a greater effect on concentrations of insulin than glucose administered directly into your veins. This effect, termed as the incretin effect, suggested signals from the gut are important in the hormonal regulation of blood glucose levels after a meal. Since that time, additional evidence has documented that digestive products of dietary carbohydrates are effective stimuli for the secretion of multiple gut peptides in the small intestines, most notably the incretins, GIP, and GLP-1. Now let's pause for a second so we can explain what exactly are these big words. Incretins is just a generalized word for gut hormones secreted by the endocrine cells that we talked about earlier in the blood cells just minutes after food consumption. They just appear there. It's a general term. GIP, on the other hand, or the gastric inhibitory peptide, is considered a weak inhibitor of gastric acid secretion, and its main role is stimulating insulin secretion. Cool stuff. GLP-1, or glucagon-like peptide 1, which we talked a little bit earlier, in the context of this topic, is an amino acid peptide hormone that is released by the endocrine L cells, or the later cells. Biochemists and biologists are thinking, what an idiot. Anyway, it's more about understanding what's going on, I think, over here than being just 100% tip-top perfect when it comes to semantics. That's just how I'm treating it, and that's what normies... I want to talk to normies, man. I know this is crazy stuff, but we can do our best. Recent evidence suggests glucose or fructose may trigger CCK or cholecystokinin release as well. Though treatment with sweet taste inhibitor, lactosol, does not alter CCK release. And the TAS1R2 component of the sweet taste receptor is not co-expressed with CCK. TAS1R2, I don't know if that's how you pronounce it, but I'm going to roll with it, is basically the taste1 receptor member 2. That's its sort of technical non-acronym name. Is a gene that recognizes natural and synthetic sweeteners such as sucrose, saccharin, ACE-K, and so forth. Aha! Meanwhile, on the other hand, we have ghrelin, which is an appetite-stimulating hormone secreted in the stomach. Its release is not driven by the luminal sensing of carbohydrates, though. Indeed, and this is very important, by the way, several reports note hunger ratings precede changes of ghrelin. Curious. Additionally, post-meal concentrations of ghrelin are not predictive of meal requests or energy content of meals. Let's talk about a study. So referenced on this is a randomized double-blind crossover study, which is a very powerful study, by the way, of 28 obese men consuming 4 beverages of the same caloric content, but one contained 50 grams of whey, fructose, glucose, or there was this fourth one which had 25 grams of whey and 25 grams of fructose in it. So 4 different beverages. Blood samples and appetite ratings were collected for 4 hours, and then they offered an all-you-can-eat Golden Corral-style buffet meal. Maybe not exactly that, but you get the point, yo. It was found that fructose and glucose-based beverages has a similar appetitive and hormonal response despite glycemic and insulinemic responses. Ghrelin, GLP-1 or glucagon-like peptide 1, and CCK or cholecystokinin after protein beverage did not affect all-you-can-eat style energy intake 4 hours later, despite the hormonal differences. Crazy, right? Moving on. Peptides are secreted in the intestines by several different kind of mechanisms. First, there is suggestive evidence for a learned anticipatory secretion that would presumably have a neural or hormonal basis. Interestingly, suppression of pre-meal GLP-1 or glucagon-like peptide 1 rise leads to lower rather than greater food intake. This is consistent with GLP-1's reported anorectic or appetite-suppressing effect, but may reflect the lack of suppression of orexigenic or appetite-stimulating signals. Secondly, glucose can pass through the sodium-glucose cotransporter 1, or SGLT1, on L cells, depolarizing the cell and releasing peptides. It's going to get a little technical here. I couldn't really make this more simple. I tried, guys. It's just couldn't. You gotta trust these words now. Anyway, the sodium-glucose cotransporters are glucose transporters that basically aid in kidney glucose reabsorption. Intravenous glucose administration does not suppress food intake acutely, whereas there is a direct relationship with intestinal glucose. This is consistent with the hypothesis that glucose transport is required for carbohydrate-mediated reduction of food intake. Third, selective nutritive sweeteners and low-calorie sweeteners can bind to T1R2, T1R3 sweet taste receptors on the endocrine cells found in the gastrointestinal tract and thereby affect the peptide release. Endocrine cells that express the receptors also possess the elements required for taste transduction. However, the terminology of gut taste may be inappropriate given the signals generated is not conveyed by taste nerves decoded by the taste cortex and does not lead to sweet perception. Nutrient sensing more aptly characterizes the function of these cells distributed among the entirety of the GI tract. Once released, these peptides may bind to receptors on different vagus nerves. Remember that nerve we were talking about earlier? This is the one. So yeah, different vagus nerve generating a neural signal or reaching the brain via circulation. This is a little bit technical. You guys can sort of kind of just get the basic drift of what's going on over here. You don't have to really understand every single thing here, all right? So, unless that's your job, then, or you're a researcher, then you can probably understand all of it. It's not a big deal. You'll be like, third graders. Identification of sweet taste molecule receptors on the intestinal endocrine cells raised the question whether low-calorie sweeteners would also be metabolically active in the gastrointestinal or GI tract. This was the case in cell culture, but support in animal models and humans has been weak. Trials have been reported no effect of sucralose on GLP-1 or plasma glucose, no effect on GLP-1, and an increase in glucose, rise of GLP-1, but with no effect on glucose. It's kind of all over the place. And an elevation of GLP-1 with low glucose concentrations. There is no resolution of the matter at this point. It is also not confirmed that nutrient signaling mechanisms identified in cell culture studies hold under physiological conditions in humans and animals. This is very important. Just because something happens in a petri dish does not mean it's going to happen in a human. Or if it does happen, the question is, how substantial is it? Should I be worried? Should I be concerned? Is this the apocalypse? Is it 2012? Did the Mayans predict it? Okay, we're getting a little bit overboard with this. Let's go back. Ingestion of high fructose corn syrup increased markedly over the past 4 decades, leading some to hypothesize that this poses a particular risk for positive energy balance and weight gain. Okay, raise your hands. How many of you know one person who thinks that everything is to blame on high fructose corn syrup? Oh, I see everybody raising their hands. Even if you can't. If you're driving your car, don't do that. Yeah. But after you stop and you're at work or you're home, then you can raise your hands. That would be cool. However, high fructose has only a comparable effect to glucose on gastric emptying, incretin release, the rate of glucose absorption, post-absorptive glucose concentrations, appetite, energy intake, and energy compensation in human. This is me adding a little bit of side note to it. I think there's a bit of a problem with all of these things, like the high fructose corn syrup, but at the same time, it's very contextual. You'll have to dig a little bit deeper to see what the problem is. Anyway, let's continue on this paper without my opinion hampering. Anyway, fructose is a sweetener. Fructose is sweeter than glucose or sucrose, so it could have a stronger rewarding properties. However, it is rarely experienced alone. The density of endocrine cells is highest in the colon, so signaling molecules are not absorbed in more proximal regions or are generated in the colon. May also prompt a robust hormone response. There's a lot of heaviness here. The concept that fermentable carbohydrates could be a source of signaling molecules that affect gut peptide concentrations associated with glucose and energy dates back to late 1980s and early 1990s. Some examples of fermentable carbohydrates we just mentioned are fruit sugar, milk sugar, fructones in bread, cereals, pastas, galactans in beans and legumes, and also sugar alcohols such as xylitol. Presently, there is considerable interest in the role of microbiota in fermenting dietary fiber to yield short-chain fatty acid, or SCFA. I'm not going to use SCFA. I'm going to say short-chain fatty acids. It just sounds cooler. So I'm going to stick with cooler. And these examples of such short-chain fatty acids include acetate, propionate, and butyrate. Just remember that these are shorter chains and low carbon, like C2C3 sort of stuff. Short-chain fatty acids influence the expression of G-protein coupled receptors and can bind to FFAR2 and FFAR3 on L cells or the later cells, resulting in secretion of peptides such as GLP-1 and PYY, which we talked about earlier. The two principal drivers of the allele break, and the allele break basically slows down gastric emptying and GI transit and also augments satiety. Sounds really cool. Allele breaks. Just imagine walking into your office one day and saying, hey, I just ate this food and I activated my ileal brakes. Yeah. However, animals lacking these two receptors have normal responses to short-chain fatty acids and improved glucose tolerance, revealing redundancy in the system and uncertainty about the mechanism. Evidence linking fermentable fiber, the microbiota, and gut peptides in humans reveals inconsistent associations between gut peptide concentrations, appetite, and energy intake. This may reflect the fact that microbiota generates many bioactive compounds that exert effects in multiple organs. Additionally, much of the work on this topic is short-term, and effects of fermentable fiber may be on growth of literally on the growth of the endocrine cells, which would reflect chronic feeding patterns. Yeah, did you think about that, huh? Maybe you did. I don't know. Fiber's impact on satiation may be overestimated, and its contribution to energy intake may be underestimated. Whether increasing fiber intake to moderate energy balance will have the desired effect is uncertain. Taken together, strong evidence indicates simple and complex dietary carbohydrates may promote the secretion of gut satiety peptides. The expectation would be that carbohydrates should have highly satiating and be consumed in limited portion sizes. However, this has not been the case in common experience, especially when it comes to beverages. This appears to belie the importance of endocrine signaling for carbohydrates in ingestive behavior. There could be three reasons why this is the case. The possibility that dietary patterns influence enteroendocrine cell density and thus peptide concentrations in the GI tract. Executive functions. Number two is executive functions such as working memory, flexible thinking, and self-control. And frequently does. This kind of stuff does override physiological signals. I shall not eat food. And I will not eat food. I'm too busy at work. Number three, no single peptide acts independently and many factors influence the hormonal response pattern to eating. Well then, whether continued exploration of carbohydrate-driven endocrine mechanisms are primarily related to only one facet of the intake, portion size, to modulate appetite and intake for preventative or therapeutic purposes will be productive is uncertain. And that's it, folks. We wrapped up carbohydrates. Oh, man. So yes, there's a lot of information here, and you will most likely have to go back to it if you want to get into the details of it. But as long as you understand just the basics of what is going on, and you can draw your own conclusions, that is a good place to be. Not everybody needs to understand every single thing, and it's in our nature sometimes. We can be really competitive. CrossFitters, I'm looking at you. It can be in our nature to just go to the end of the world, but the goal here is abstraction. And even I, myself, I've read this paper. I understand a lot of this stuff. Even I don't remember every single thing. So let's be humble about it, and let's not go to the extremes of it. And just try to take practical takeaways over here while at the same time learning a little bit more step by step. You have a life to live. You got family to feed. You got jobs to do. That's your highest priority. This is just extra stuff that can help with your health and your experiences in life. Just think of it that way, just in a very generalized way. Anyway, let's move on to protein. And so the endocrine responses to the protein. In 1970, it was demonstrated that sauron attacked... never mind. That's another page. In 1970, it was demonstrated that intake of midday and subsequent meal were lower when the earlier meal was higher in protein. Subsequent studies in humans supported these findings. In addition to decreased energy intake, self-reports of hunger and desire to eat were significantly lower in high protein compared to high carbohydrate meal. Over here, we have a reference study in 1986. In a world on Earth. In 1986 study with 13 healthy male and female subjects whose ratings of hunger, appetite, and fullness were measured 5 minutes after and 1 hour after two same calorie meals, and the one meal was about 31% of total energy from protein, and the other one was 52% energy from carbohydrates. In this study, it was concluded that subjects showed preference for carbohydrate-heavy foods and aversion towards high-protein foods, disregarding what meal they ate. Yeah. Hypothesis for the increased satiety due to protein, including circulating amino acids, we discussed it in the last one, increased thermogenesis with protein intake. So this was basically the aminostatic theory. And then, of course, there's also some people thought it was the sensory-specific satiety, and they also thought that it was the gut hormone release. So these are different ways people try to explain why protein is so filling and satiating, and it kind of works. Now, but as over time, the associations between protein exposure and gut peptide release are weak and inconsistent. This stems in part from influences of quantity, quality, and type of protein ingested on gut peptide secretion. Research into the effect of quantity of protein consumed on appetitive peptide concentration began in mid-2000s. Findings were suggestive of positive effects, but not compelling. In 2013, a study with 25 men participated in a three-way randomized double-blind crossover study. That's pretty intense and powerful study. Test meals were 30% from fat and protein, adjusted at the expense of carbohydrates. Meals were isocaloric, some calories with 14%, 25%, and 50% protein, and then followed by an ad lib all-you-can-eat Golden Corral-style buffet 4 hours later. I should change this from Golden Corral to some other buffet place. Leave some comments and let me know where else you can go for buffets. I like after COVID, I haven't been eating at a buffet. It's been years since I've been eating at a buffet. Anyway, so much tangents. God, I'm the worst. Protein in this study increased GLP-1, PYY, and glucagon dose-dependently. So all this stuff went up. Satiety and fullness also increased dose-dependently. Hunger and prospective consumption decreased dose-dependently. So all this stuff was good. Ha! Here's the kicker. There was no effect on GIP and CCK, ghrelin, or reduction in the ad lib energy intake when they went to Golden Corral's all-you-can-eat buffet. It's really not all-you-can-eat Golden Corral buffet. It just means that it's an ad lib energy and intake sort of buffet. I'm not sure exactly what was in it. This makes sense because people always eat to fullness. And look at the obesity and overweight rates in USA and all over the world. This is literally the problem. People always eat to fullness. And then stuff doesn't go their way. They become overweight or obese or diabetic, and all these other things start happening. So that is the problem. Just because you feel full doesn't mean you're going to be in a net caloric deficit. Just because you ate something and it makes you feel full doesn't mean that thing is responsible for making you thinner or fatter or whatever the case is. Anyway, furthermore, a review of acute feeding studies in humans noted that high-protein interventions led to lower hunger, ghrelin, and increased fullness in only 35%, 37%, and 55% of trials, respectively. In other words, the sensations themselves were not reliable. You cannot trust your own sensations, my friends. They betray you. They were not the chosen ones. GLP-1 or PYY concentrations of high-protein groups were elevated in only 47% of trials, and food intake at a challenge meal was lower in only 18% of the trials with a high-protein intervention. According to the authors of this specific review, the one that I'm reading and literally paraphrasing and reading off of, they say that these inconsistent results may be due to limitations of preload design protocols. Preloads are basically the foods that you consume as the test meal, and interstudy differences in protein intake levels. So they just fluctuate in these studies. Weakness of preload paradigm include assessment of laboratory-based atypical or irregular eating behavior, variability of test meals, both composition and timing, inconsistent challenge loads, short time window between eating events, and fixed meal times instead of meal requests. These studies also manipulate protein loads relative to meal composition. Example, percentage of energy, percentage of weight, and consumer characteristics such as energy need, percentage of body weight. So these things hamper the isolated effect of protein, if there are many. I'm going to use the word any. That's a good one. Protein sources and quality have also been explored as predictors of satiety, reportedly mediated by gut endocrine cells. Results from these studies are numerous and inconclusive. In 2 randomized single-blind crossover studies, see, I like the good studies. I like these crossover blind ones. I like these. It reminds me of Daredevil. Yeah, it's a bad joke. Anyway, in 2 randomized single-blind crossover studies, it was noted that casein and pea protein had an impact on food intake compared to whey protein. However, this effect was noted only when protein was consumed as a starter and not 30 minutes before the meal. I believe even straight-up water does that too. Like straight-up water. Yeah, this water part was not part of this review paper, by the way. I just added it. In another study, 30 fasting, non-smoking premenopausal women attended a research center on 3 days to watch Daredevil. To consume breakfast meals containing pork, beef, and chicken, all matched in calories, protein content, palatability, and appearance. No difference was found in energy intake or macronutrient profile of food at the ad-lib buffet lunch or over the rest of the day. Hunger and satiety over 180 minutes period did not also differ between the test meals. Hence, no difference on satiety or appetite-related hormones or insulin. What a tragedy. Additionally, different protein types and combinations of proteins result in variable peptide release. Ghrelin secretion notably varies with protein source. When the protein is controlled, dairy, plant, and animal protein led to a reduction to a lesser extent, and increase in plasma ghrelin, respectively, though appetite ratings remained the same or similar. Similar is a better word. You gotta be picky with these scientific terms, man. You can't just be same or similar, but please, I beg forgiveness. For the sake of simplicity, I sometimes roll over these terms. And to make things fun, a little bit of entertainment and laughter goes a long way. Laughter makes the most evil-looking face friendly. That's my philosophy in life. Effect of whey and casein are frequently contrasted because whey is digested and absorbed more rapidly than casein. They do, casein. They're both milk proteins, though. Whether these digestive peptides directly stimulate appetitive peptide release or whether the digestion rate of dietary protein modulates the release is uncertain. Liquid whey preload reportedly leads to significantly lower energy intake, increased fullness, decreased desire to eat, and elevated GIP, GLP, and CCK compared to an isocaloric casein preload. So casein's a slow digesting, weighs faster. Just throwing it out there. The increase in CCK with whey preloads has been attributed to the 64 amino acid peptide product of whey digestion called caseino-macropeptide or CMP. That's what they thought. So what they're trying to do is take protein sources, like for example, milk, and they're trying to further isolate and figure out where the link is, which is causing this so-called satiety effect on protein. So they're further refining. So they come down to caseino-macropeptide. Early research with rat models indicated CMP or caseino-macropeptide stimulates release of CCK. However, subsequent human studies found CMP alone or caseino-macropeptide alone or with an energy-containing preload has no effect on subjective satiety, CCK release, or food intake. Well done. A review on the satiating effect of dairy proteins concluded whey protein stimulates secretion of GLP-1 more than other proteins. But there is no clear evidence CCK, PYY, and ghrelin concentrations are affected by protein sources. Discovery of endogenous satiety peptides led to experimentation with the exogenous administration of synthetic forms of these peptides that we talked about, CCK, PYY, ghrelin, and so forth. In these administration of peptides reduced energy intake and or increased satiety, feelings of satiety. However, a meta-analysis. So meta-analysis is just they'll take a bunch of studies, all of them together, lump them up, and be like, hey, what's going on? You came up with one study, you came up with another. Let's look at all these 25 different ones and see what's happening. Are they all predicting the same thing? So that's what a meta-analysis is. And henceforth, a meta-analysis examining the difference between exogenous concentrations and endogenous productions of PYY, GLP-1, and CCK found concentrations of exogenous PYY and GLP-1 that elicited a satiation response were significantly above physiological concentrations, leaving open the question about their nutritional relevance in humans. So they came up with results when it comes to PYY and GLP using values that are not even possible using normal food. It's impossible unless you just seriously just inject it. That's how you get the energy intake decrease. This is the problem. I was talking about in the last episode, and I was talking about the processed food episode. Like, how realistic are these interventions? Are these yardsticks even reliable or practical? So there you have it. There's the problem. It's not physiologically possible, at least based on this review. Macronutrient composition was not evaluated for the above meta-analysis, limiting the ability to determine the effect of protein on satiety peptide levels. So there's that. There's an issue with this as well. So you gotta look at the back and forth. You gotta do to and fro, up and down, left and right. Bonnie and Clyde. No, those were synergistic. Jekyll and Heidi. Haha, that's a good one. Whether gut peptide hormones are released primarily to signal satiety or to regulate digestive processes, primarily with appetitive sensations, only associated is receiving greater consideration. The effect of one macronutrient like protein and release of any one or combination of appetite peptides is unlikely to be the primary driver controlling energy intake. So you're starting to see the picture here. That this is a symphony. This is more than just one macronutrient to rule them all. This is not Lord of the Rings. This is a lot more than that. It's a government. It's an institution. It's a corporation. It's a group of people that create something. So that's what it is. Please don't politicize it. I'm just an innocent food guy. All right? Let's move on to the fat. Endocrine responses to fat. A link between lipid consumption, gut peptide secretion, and satiety was reported as early as 1937, when an extract from intestinal secretions collected after an olive oil dose suppressed feeding in rabbits. I wish I had rabbit as a pet. How many of you actually have a rabbit as a pet? Please comment. What's your favorite pet? All right, we're kind of sidelining the podcast. Let's go back. Because later research documented that dietary lipids are potent stimuli for release of peptides such as CCK and PYY, some researchers hypothesized that lipids are primary regulators of energy balance. We talked about the lipostatic model in the last podcast, so we're moving on to a little bit more, and we're moving to the endocrine now. The endocrine mechanisms by which dietary lipids reportedly influence digestive behavior and digestive processes are mediated by signals that begin in the oral cavity, that is your mouth. In rodents, oral fat exposure reduces the variability of gastric emptying, or the rate at which food leaves your stomach, under conditions of different load rates, and increase pancreatic and endocrine secretions. In humans, oral fat exposure elicits gastrin, gastric acid, and ghrelin secretion and enhances gastric emptying. Additionally, oral exposure stimulates the release of gut peptides, example, CCK, and GLP-1. Man, swear, we're having an abusive relationship with GLP-1. These products are also released by the detection of medium and long-chain fatty acids in the small intestine in dose-dependent manner. In one study, an intestinal infusion of C12, 12 carbon chains, which is sort of like a longer chain, but not C10, sort of like closer to medium chain, or control significantly reduced desire to eat and hunger ratings, though it did increase nausea because of medium chain fatty acids. Yeah, you can't really eat a lot of oil on its own, man. May the gods who chug down butter bestow us with their blessings for doing the impossible. Or olive oil or any of these fats on their own. Disgusting. You got to use it with something else. That's why we created the energy pods. This corresponded to male participants consuming 2,857 kilojoules less energy at a single buffet meal compared to control. Some studies showed that long-chain fatty acids are even more potent stimulators of gut peptide release, as PYY secretion responds in proportion to increasing chain length of fatty acids, and CCK, or cholecystokinin, concentrations increase more in the first 30 minutes after ingestions of long-chain fatty acids compared to medium-chain fatty acids. Even so, there appears to be no differential effect of fatty acid chain length on hunger ratings or energy consumption in lean subjects. The reference study is a 2010 study using short chain dairy fats, medium chains from coconut oil, and long chains from beef tallow test breakfast containing 52 grams of fat in a randomized crossover study with 18 lean men. There was no detectable difference in visual appearance, smell, taste, and aftertaste, and palatability, and no significant effect of fatty acid chain length on hunger, fullness, satisfaction, or thoughts on the food. After the test meal, 3.5 hours later, a buffet, Golden Corral, buffet was offered, and no differences were observed in energy or macronutrient intake. Maximum difference was 2%. Ouchies. Anyway, while not a result of dietary fat intake, short-chain fatty acids are generated by the microbiota through fiber fermentation and also stimulate secretion of gut peptides such as PYY and GLP-1. God damn it, man. GLP-1. However, administration of acetate, which is C2 to 2 carbon, very short chain, and propionate, which is 3 carbon, very short chain again, does not reduce energy intake. Indeed, short chain fatty acids are a source of energy, 8 kilojoules per gram, not calories, please note, and can increase one's energy balance by an additional 150 kilocalories per day through energy harvesting done by the microbiota, your microbiome. So there's that. The effect of fatty acid saturation on secretion of gut peptides and appetitive sensations is also not established. Some work suggested that the consumption of meals high in saturated fatty acids leads to lower hunger and greater fullness scores compared to loads containing MUFAs or PUFAs, monounsaturated fatty acids, and PUFAs, which is polyunsaturated fatty acids. While other work indicates that there is no significant association between fatty acid saturation and either gut peptide secretion, appetitive sensation, or food intake. So this is one that's going to sort of sting. This is kind of crazy, isn't it? Even when differences are noted, energy compensation in the period following the test meal consumption offsets the reduction of energy intake, which indicates the effect of fatty acids and gut peptides on energy intake may be very small, or small, as the author said, not very small. Interpretation of these literature relating fat intake to appetitive sensations and diet is difficult to assess due to differential inter-individual responsiveness. For example, sex and body mass index may affect the gut peptide response to unsaturated lipids. While not studied, almond oil containing mainly oleic acid, which has a lipid number of 18.1, increases CCK concentration and decreases hunger ratings in females but not males. And duodenal infusions of lipid composed primarily of long-chain unsaturated fatty acids did not increase CCK and GLP-1 in males, nor did it result in any difference in energy consumption. So it's really muddy out there when it comes to all these different mechanisms. An experiment with a similar experimental paradigm and a more aggressive dose scheme observed increased gut peptide concentrations, but still no difference in energy intake in treated males. Additionally, adiposity may be an important determinant of responsiveness, as an intestinal infusion of oleic acid increases CCK concentrations to a greater degree in lean as compared to obese individuals. And ghrelin concentration in obese males do not decline as much after a meal as in lean males following administration of long-chain fatty acids. Taken all of this together, the evidence suggests dietary lipid stimulate secretion of gut peptides reported to hold satiation, satiety effects, but they have limited impact on long-term energy intake. There are several reasons for this. First, they act primarily on meal size, but generally not on eating frequency. Indeed, high-fat diets are often associated with positive energy balance and weight gain. This has been attributed to tolerance or adaptation of the GI response with chronic consumption of high-fat diets. Second, there may be certain subgroups of responders, possibly related to sex or BMI, and gradations of responsiveness within the category without a dominant pattern. Third, the range of lifestyles and conditions within a population may make certain gut peptides the primary determinant in particular situations, but not in others. And fourth, the physiological systems may override satiety signals from the gut peptides. Thus, under customary dietary conditions, the consequences of intestinal lipid signaling and gut peptide secretion on appetite and energy intake is limited. So this was a lot to take in. We've just concluded fat as well. And you don't have to just absorb every single thing that was mentioned in this paper and just take what is relevant. Perhaps we'll give this another listen as well. And yeah, I understand this was a very loaded podcast. There's a lot of information that got thrown out there. And next week is going to be even more insane when we get into the brain, the neural response. Even I'm dreading how I'm going to do that show. But luckily, after this podcast, after the neural podcast, we're going to have a podcast with someone like Danny Vega. And we're going to have a little bit of a chill time after the science bombardment. And I think we all deserve it. We need a break from the science after that. And we're going to have a good time. We're going to talk about Florida life. We're going to talk about life and lifting. Because that's all what life needs to be. Energy pods and lifting. Yeah. Or you know, whatever you want to do. Just forge your own path in life and enjoy it. Science is a means to an end. And your journey in life, we have to strive to become better human beings every day. And knowledge is just one facet of it. It is auxiliary. It is not the centerpiece. You are the centerpiece of your own life. Write your own narrative. Paint a picture on your own canvas. That's the most important thing over here. Hopefully all of this information will help you, and hopefully you got some takeaways from it. I know this was a lot, and but we have to go through this journey. This is really important because this is a very widely targeted scattershot, and you guys know we have been at the heart of keto, low carb, and a lot of different health communities. But I wanted to speak the truth. I know it sounds cliche, but I wanted to be honest, and honesty requires you to falsify and critically look at everything that you're doing. And that's why this paper really, I got attracted to it, because my goal is to help and solve problems. And we do that by exploring and thinking rather than dogmatically staying on to our beliefs. It's easy to be in an echo chamber. It's easy to join a cultish attitude towards something, but it takes courage and mental resilience, I think, in my humble opinion, to go out there and actively hunt for something that can help humanity. And this can be a very humbling experience and takes you outside of your comfort zone and even outside of your comfort circle. Forge a path. Take this. If there's anything you can take from this podcast, just beyond just the science, is just try to forge your own path. Try to savor life and never stop learning. Whenever you see something that screams at you, and it's the easiest path is just to adopt that. Step back and just think that there's always two sides to an argument. There's always somebody else saying something else, and you got to ask the question, what does the other person think? That's what has kept me as a human being safe through some of my hardest journeys in life. And I know this is getting a little bit more philosophical, but there has to be a philosophical note to it because, science, it doesn't draw a philosophical image. It doesn't create an ideology. It just is. That's what science has been. But what we can do is take this tool and further use it to improve our lives. So thank you so much, guys and gals, for being on this solo podcast with me. I don't normally do these, but I would love to know, whether through email or through your comments, whether this was a good show and I did well. I know I'm not a teacher. I've done some TA stuff in my college, and biology was never one of the things that I delved deep into before Ketogeek. So all of this stuff is new, but it so intrigues me and interests me. I really just have to go into it. And everybody who's been here listening to this end of this show, you guys are amazing human being. And above all, thanks to all the customers who have been with me through this journey and put their trust, bought our products. This knowledge could not have gone out without your support. And above all, I would like to still thank the writers of this paper. I'm saying it. You just, like, all of this knowledge, I would say 99% of the credit goes to them. Think of this as their words, and me just trying to compile this so that it's more understandable. I wish I wasn't the person doing this, but I have to do it because, you gotta talk about the truth. You gotta talk about actively falsifying yourself, and I'm doing the best what I can with my resources that I have and the way I can think. And if anybody has more who wants to, like, I know there are people who are scientists, but the most important thing on this podcast, I want to focus on is, yeah, you got the science, but how do you want to give back to the world? How do you tie this into a greater vision for a better future, better humanity? Look, we all have to die 6 feet under at one point in life. And this is normally stuff I wouldn't say when Corey was around or any guest was around, but we all have to go to the same destination. The big question is, what do you leave back for this world? That's the big thing. You got to leave the world back a better place than you found it in. That's so important, I think. I know this is not something that we think on an everyday. You got your work, you got your job, you got all these things going on. But at the end of the day, you got to have some anchor, some anchor of this, so you can go back to it. And I think that leads to a more fulfilling life as well. But let me give credit to the authors of this paper. I'm not good with names. I'm really trash with names. When I was, English was not my first language. I know it's hard for some people to believe, but many of you can probably tell. But I was raised in Pakistan, so my English has evolved rather than be there in the first place. But the authors of this paper are Alicia L. Carrero, Japna Dillon, Susanna Gordon, Ashley G. Jacobs, Kelly A. Higgins, Brianna M. MacArthur, Benjamin W. Redon, Rebecca L. Rivera, Leah R. Schmidt, and Richard D. Mattis. Thank you guys for writing this amazing review paper. I wish I could have had one of you people show up and we could talk for hours about this, and especially what new things you guys have been learning from writing this paper, because this is a 2016 paper and we're in 2021. So the best thing you guys can do for us, at least, is go buy our energy pods. They're going to be available on Amazon, not Prime yet, on this coming Thursday. But you can always go to our website, go buy them, or you can subscribe to them on a monthly basis. This gives us a constant revenue towards shoving some money towards podcasting and our information section. And what we also do is when we have a more surplus of money, we throw it towards certain events as well that promote better science and research and education. We've done that with Low Carb USA and some other things as well. So, but we want to obviously branch out to beyond just low carb as well, as you guys can probably start seeing the inevitable evolution of Ketogeek. So let's create a better world, and eventually, I think my goal with energy pods is to find ways that we can come up with better products. And right now, let's be honest. I mean, I understand energy pods are something that not a lot of people can afford, especially when you look at different countries across the globe. These are expensive products. They're not cheap, and almond flour is something that is a bit of a luxury item at times. But what we're aiming for, you and I, listener and customer and myself, what we're aiming for is something a little bit bigger than that. Just take this investment and put it back into this world that we live in. I think that's the bigger picture here, and we have to work together in this world to create a better future. As cliche as it sounds, that's just the hard truth. So thank you so much for being on the show. And while you're at it, just leave us a review too. It takes about a second to just click one of those stars, and thanks for all those people who sent positive feedbacks. And just when I blasted that newsletter and the sales started rolling in, I was like, okay, these guys are amazing human beings. They still are with us at Ketogeek. I know we took one and a half year off. We went a bit of a silent mode, but we're back. And as you can see now that we have quite evolved beyond where we started, and we will continue our journey forward. So thank you so much, and see you guys next week. Thank you for listening to the show. You can visit us at Ketogeek.com to check out our premium keto-friendly and low-carb food products, including our infamous energy pods, made from the best ingredients we could find in the entire country. We also recommend signing up for our newsletter at Ketogeek.com/signup, where you get exclusive discounts, rewards, product announcements, and the latest content we are working on. You also get a 10% discount on your entire order when you sign up. Go to Ketogeek.com/signup to join the low-carb playground.